Thanks. So I'm really delighted to be here today. This has been really an interesting symposium that's covered so many different aspects of the disease. So today I'm going to tell you a little bit of a different story about our efforts to use high throughput screening to identify vulnerabilities against SARS-CoV-2 and the platform that we've established to look for antiviral therapeutics that could potentially be repurposed for use during this pandemic. So I want to start by talking a little bit about antivirals and the classes of antivirals that are out there and the classes that we've been focusing on over the last year. So most antivirals that are in clinical use are direct-acting antivirals. So those are small molecules that target essential viral proteins to block the ability of the virus to replicate within cells. So a good example of that is Remdesivir, which is a nucleoside analog that blocks the RNA-dependent RNA polymerase activity of this RNA virus and therefore can block replication. In addition, because viruses are obligate intracellular parasites, they have to use host pathways and machinery in order to replicate in cells. And so there's been a lot of effort to develop host-targeted antivirals that will block one of these essential functions that are required for the virus, which then could ultimately block viral infection. And so a good example of this are entry factors, which have been identified. And the idea is if you can block the entry of the virus by blocking some essential component within the cell, you could then again block viral infection. And this would potentially be a useful therapeutic. In addition, there's another class of host-targeted antivirals, the immunomodulators, which are really of great interest. And these fall into a number of different classes too. So some groups are working on boosting immunity early, and so these would be the innate modulators, and other groups are working on trying to ameliorate the pathogenesis associated with hyperinflammatory states later in infection. And so there's a lot of efforts to, again, modulate host pathways within the immune space. But today I'm going to focus on talking to you about our efforts to identify human proteins that are essential for infection and antivirals that may target them. Okay, so in thinking about host-targeted antivirals, the idea is you could try to find vulnerabilities that block some step in viral infection. And then we need to understand how these host factors or host-targeted antivirals impact the replication cycle of the virus. And so as is obvious, some of these will impact signaling pathways within host cells that are essential for infection. Others will target metabolic pathways within cells that are essential for infection. And it becomes challenging to uncover how these antivirals are working. And so we've been doing a number of downstream analyses using omic approaches to understand how these antivirals may be functioning using RNA-seq and proteomics, in addition to other assays that maybe we'll talk about later. Okay. So again, these host-directed antivirals block infection, but it's not always straightforward to figure out how they're impacting viral infection. And it's really important not only to find antivirals, but to understand how they work. Okay, so the screening platform that we have here at Penn is based on a high-throughput screening core that I'm the scientific director of. And so what we have here is a facility where we can use high throughput screening to identify antiviral molecules. So we use 384 well plate formats where we can seed human cells, and we'll talk more about the cell types of interest here. And then we use robotics and liquid handling to add the drugs to the wells. And then we take these plates into the BSL-3 that we have here at Penn and infect them with the virus. And then we use downstream analytics to identify the antivirals. So the assay that we use most routinely uses automated microscopy, where we can look at each well using automated image analysis for the amount of infection in each well. And so what we do here is we can look at in wells, and we're looking for the blue signal, which is staining the nuclei. So we can look at the number of cells in each well, and then we can stain with a viral antigen to look at the amount of viral infection in each well. And so what we're looking for are antivirals that block infection but don't affect toxicity. So the same number of blue in each well, but a decrease in the amount of green, the amount of virus. And so this is the platform that we've been using to try to identify antiviral small molecules. Okay, so what libraries are we screening? So we've sort of stepped through a number of different libraries that I'd like to tell you about, because the libraries are obviously important in defining what you may identify. So since last March, we've been scouring the literature to identify antivirals that are reported to be active. And we've purchased a large number of the antivirals. Basically every week we call the literature to test things in our own assay platform. We also, in the high-throughput screening core here at Penn, we have our own in-house repurposing library of about 3,500 drugs, which include all the classical small molecule inhibitors of kinases and epigenetics and other sort of classical targets. That includes about 1,000 FDA-approved drugs and another 1,000 clinical trial drugs. We've also partnered with Medicines for Malaria Venture, a non-for-profit global organization. And we did that early on because they had many chloroquine derivatives. And as you know, there was much interest in hydroxychloroquine early on. And so we wanted to test as many chloroquine derivatives as possible as fast as we could. We also partnered with NCATS, which is the NIH screening group. So they have their own repurposing library, and we basically got from them another 2,500 drugs that were not in our repurposing library in order to screen for antivirals. And then more recently, we partnered with the Gates Foundation and Caliber to screen their library, which they call Reframe, which is 12,000 drugs that have been in at least phase 1 clinical trials. And so together, this is about 20,000 compounds that we've screened against SARS-CoV-2. Okay. So our pipeline that we've implemented over the last year is to basically take these high throughput screens where we have these repurposing drug libraries, about 20,000 compounds. We've screened them in a variety of cell types that I'll tell you about in a minute. And we've done all of those compounds against SARS-CoV-2. But as you know, there are many coronaviruses out there, including a number of circulating coronaviruses. And so we're interested in identifying drugs that might be pan-anticoronavirus. And so we've been screening now the libraries with OC43, which is one of the low-path coronaviruses. So once we find, using the single point screening platform, once we find our candidates, we then repurchase the powders and perform validation using 8-point dose response curves. And we've been doing this in a variety of cell types, again, to identify drugs that are active in the most relevant cell type, for example, in the respiratory epithelium, but also looking at other cell types, because again, the best drugs would be drugs that would be more broadly active. And here we're doing 8-point dose response, looking both at infection and at toxicity. Again, we're really interested in drugs that are non-toxic and have high activity. So these assays use a microscopy-based platform, and so we think it's really important to use an orthogonal assay to validate not only that they're active, but to look more carefully at the magnitude of inhibition. And so we use RT-qPCR assay, which gives us a great dynamic range to look through the drugs that we identify for those that are as antiviral, at least as Remdesivir, which is about 3 logs. So a number of the drugs that we've identified have at least a 2 to 3 log decrease in viral infection. So once we find candidates that are active in these assays, we then want to further characterize them. We want to understand the mechanism of how they impact SARS-CoV-2 infection. Are they impacting entry? And we have assays for that. Do they impact other respiratory viruses, other coronaviruses, and how do they impact the RNA or proteins within cells? And can we use genetics to understand the mechanism of action? Then, of course, these assays are done in cell lines. It's very important to validate that any candidates that we identify are also active in primary respiratory cells. And so the airway epithelium is a stratified epithelial layer, and that can be modeled using a variety of tools. But the platform that we think best reflects the human respiratory tract are these air-liquid interface cultures, which we basically, by removing the air, these cells differentiate into a variety of cell types within the culture system, where they become very ciliated, they beat, they make mucus. And at this point, they become very permissive to SARS-CoV-2. And we've been basically using this platform looking at the nasal, bronchial, small airway epithelium in collaboration with a number of different groups to look to see if the drugs that we identify are active in these more relevant culture systems. In addition, because there's a great core at Penn, an IPS-derived core, we have also been testing some of our drugs in a human IPS-derived AT2 cell model, which can also be quite useful. Okay, so then once things pass this hurdle, we then are moving them into preclinical testing in a variety of small animal models, and we're collaborating with Mike Diamond's lab and Ben Tenover's lab to test many of our candidates in these animal models, with the idea of once the candidates pass this, we then potentially would go into a clinical trial mode. So it's really, there's a number of steps that we're using to really prioritize candidates to move forward with. Okay. So this is just some data from our earliest screens that we performed last year when we were getting the assay platform up and running. So we were started with this 3,500 drug library that we had within Penn already that we were ready to go with. And so the first thing we had to do was find cell types that were permissive to infection and to sort of optimize the assay parameters so that way we could identify candidates. So we started with this African green monkey cell line called Vero, because actually that's the cell line that we grew the virus in. So while we were growing virus, we could very rapidly develop an assay to use these model cells. And in screening these 3,500, we only found a very small number of drugs that were active and that validated in our dose response assays. While we were doing these assays, we really wanted to find human cells that were permissive to infection for obvious reasons. And the first human cell line that we identified that was permissive was a human hepatocyte cell line called Huh7.5s, which are actually very well established cells for studies of hepatitis C virus infection, and they're very permissive to many viral infections. So we rapidly were able to infect these cells, and we performed a parallel assay looking for antivirals, where we identified a larger number of antivirals that were active in these cells compared to these cells. But, given this sort of lack of concordance here, let's say, we became interested, obviously, to look in the respiratory tract. And so we obtained a large number of cell lines from a variety of sources to look for human respiratory epithelial cell lines that were permissive to infection. And the first one that we found that we became very interested in was a cell line called Calu-3, which is being widely used in the coronavirus field because they're a very good model for respiratory epithelial cells. And so we took these candidates from these screens and asked, well, how many of them were active against the SARS coronavirus infection in Calu-3 cells? And of these candidates, we found only 9 were active in the Calu-3 cells. So the majority were not. And so that became a little bit interesting to us, and I'm going to talk more about that. But the 9 that were active in at least 2 of these cell types fall into these classes here, and we'll talk more about them in a minute. But I wanted to first mention something about the category here and how that really led us to understand more about the biology of infection. So one of the candidates within this group was hydroxychloroquine, which is active in these cells, but did not seem to be active in the lung epithelial cells. And so I just want to show you what that data looks like because I think it's very striking. So as I said, Remdesivir is an RNA-dependent RNA polymerase inhibitor, and it blocks infection in all 3 cell types. So again, cell viability is shown in green, so it's non-toxic, but it can completely block infection to varying IC50s in the different cell types. Okay, so this is how the data is going to look going forward. So if we treat Vero cells with the hydroxychloroquine or a Cathepsin inhibitor, which is a protease that's acid dependent, and so this protease has to be active in order to cleave spike to allow fusion, you can see we can block infection very nicely. Whereas an inhibitor called Canistat against TMPRSS2, which is a surface serine protease that had been recently shown to be active in some lung models, had no activity in Vero cells. And that's because Vero cells don't even express this. So Vero cells are permissive in the absence of this molecule and use this acid-dependent protease pathway to trigger spike. Similarly, in these hepatocyte cells, we see exactly the same phenotypes where there's an acid-dependent protease activity that's independent of TMPRSS2. But in contrast, if we look in these Calu-3 cells, this lung epithelial cell line, what you can see is hydroxychloroquine has no activity, the Cathepsin inhibitor has no activity, but the serine protease is absolutely essential. So you can block infection using Camostat, and actually what you can see is actually the cells now continue to divide because they're so protected from infection. Now they're growing during the time course of this assay. So this is the kind of drugs we're really looking for, are drugs that are active against the virus and have no toxicity. But this again points to the really fundamental differences between what's going on in these two cell types, where the virus can actually use independent pathways for infection. In some cell types, it's completely dependent on this cathepsin mediated activation, whereas in other cell types, exemplified by the Calu-3 cells, we see that this pathway is completely dispensable, but the virus is dependent on this TMPRSS2 mediated triggering. So again, we've looked at other respiratory cells, as I alluded to earlier, that we can look by qPCR, and you can see in Calu-3s, the human iPS-derived AT2 cells and the air-liquid interface cultures, Camostat can block infection, whereas this pathway doesn't seem to be heavily engaged. And the fact that the virus can use multiple pathways may suggest that this is not perhaps the best strategy for blocking viral infection, because in vivo, we don't really know the full spectrum of which pathways are being used in which cell types under which conditions. So to get back to the antivirals that we did identify that were active in both hepatocytes and the lung epithelial cells, they're shown here. So 4 of these are approved drugs. 4 of these are actually in clinical trials. We identified a number of receptor tyrosine kinases and other pathway components that we'll talk more about later. But I want to talk a little bit about cyclosporin here because we found this very interesting early on. So cyclosporin is a very known immunosuppressive agent. It's used broadly globally, and how it works is it blocks cyclophilin and calcineurin, and calcineurin is required to activate the transcription factor NFAT that leads to immune activation. And so in some viral infections, cyclophilin is important for infection, while in other viral infections, activation downstream of calcineurin is important for viral infection. So we're interested in understanding how cyclosporin might be antiviral against this virus and what the consequences of that are. So here you can see in 3 different cell lines, this hepatocyte cell line, this lung epithelial cell line, and then this other lung epithelial A549 cell line that has a transgene expressing ACE2. You can see in all these cell types, cyclosporin is active to block infection. We were interested in other coronaviruses, as I alluded to, and whether or not things were pan anti-respiratory virus. And so you can see here, we could block infection with OC43 treating with cyclosporin, but it has no impact on influenza, suggesting that this pathway is important for coronaviruses, but not every virus that we test, suggesting their specificity. So we did a number of downstream experiments where we inhibited calcineurin directly, or NFAT, and that did not affect infection, suggesting that the activity is through the inhibition of cyclophilin. And in order to fully address that, we tested a drug called Alisporivir, which is actually an oral cyclophilin inhibitor that doesn't target calcineurin. And it's been tested in clinical trials against hepatitis C virus infection, where it was added to the current regimen at that time, and it didn't show any increased boost of activity, and so it was not further explored. Nevertheless, it didn't show terrible side effects. So we showed that in Calu-3 cells, again, we could inhibit infection using Alisporivir, as well as in primary human bronchial epithelial cells and these iPS-derived type 2 alveolar cells, suggesting that indeed blocking this pathway could ameliorate replication in a variety of contexts. So there's a number of clinical trials ongoing with cyclosporin, including one at Penn. While the prospective trials, I haven't seen any of the data that's come out of them yet. In a retrospective trial that recently came out of Spain, this treatment showed benefit, suggesting that perhaps both targeting the immunopathology as well as viral replication might be beneficial to hospitalized patients. In addition, our data suggests that we might consider Alisporivir in acute infection, given that it can attenuate infection, although we haven't yet tested this in animal models. Okay. So given the fact that we saw such big differences in sensitivities between the different cell types, it became really clear to us that we should basically go back and do all of our primary screening in these lung epithelial cells, the Calu-3 cells. So this is basically pie charts showing you the screening data from the 3 different libraries that we've screened against SARS-CoV2 and Calu-3 cells. So we screened our 3,500 drugs from Penn, we screened the NCATS library of 2,500 drugs, and we screened the Reframe library of 12,000 compounds. And we identified a number of different candidates from these 3 different screens. And what I've done here is I've shown them as pie charts, where the pie charts are colored the same across the 3 different screens that we ran. And so obviously we're characterizing these candidates, but I wanted to highlight some of the classes that we identified and how we're moving forward, and where I hope that we can go soon to see if any of these would be able to be repurposed against SARS-CoV-2. Okay, so one group, of course, are within this Reframe library, there were a number of different TMPRSS2 inhibitors, and I showed you earlier slides that showed that Camostat had very nice activity blocking viral infection. So given that, we were interested in whether or not there were additional inhibitors that behaved like Camostat to block the entry pathway within these lung epithelial cells. So the question was, are some of our candidates entry inhibitors that block this pathway that's active within these lung cells? And so we basically employed a recombinant vesicular stomatitis virus setup in order to ask this question. So we obtained from Sean Whalen both VSV virus and VSV that is recombinant and carries the SARS-CoV-2 spike protein. So the idea is anything that's spike dependent or entry dependent will inhibit these viruses, and anything that's specifically replication of SARS will affect this and not this. And so you can do the systematic testing to find things that are specific for entry. So just to show you, if we treat cells with Remdesivir, we can block SARS infection, but it has no effect on VSV replication, which is known because it doesn't inhibit the RNA-dependent RNA polymerase of VSV. Likewise, if we treat with Canistat, we can see a block in infection with SARS-CoV-2 and with the VSV that carries spike, but it has no effect on VSV that doesn't carry spike because it doesn't use this TMPRSS2 dependent entry pathway. So basically we screened all of the candidates that came out of any of those screens. And one of the candidates we found is this drug called UK371-804, which is actually a serine protease inhibitor that was not known to target TMPRSS2. And so this suggests that we've now found another TMPRSS2 inhibitor that, that can block entry. In addition, we found that 4 other additional compounds that seem to target this entry pathway, and most of them are known activators of PKC. And so we're interested in how activation of this protein kinase C can impact this entry pathway. But nevertheless, we now have a very systematic assay system to stratify hits into entry or downstream of entry.
Sarah, this is a 2-minute warning.
Okay. So we also identified a number of nucleosides and nucleoside metabolic metabolism antivirals. And so just to show you, we identified not only Remdesivir, but the new Merck drug molnupiravir and additional nucleosides that are active in Calu-3s. And just, entry isn't the only pathway that's really different between different cell types. If you take the same inhibitors that are targeting the RDRP, you can see that there's differences in sensitivity across these different cell types. So really many different facets of the replication are different in different cell types, making it really important for us to study infection in the cell type that we think is most relevant to infection. And we're screening additional nucleosides now. So within the nucleoside metabolism category, so this is a busy slide, but this is a metabolic pathway that leads to the de novo synthesis of different nucleosides that are of course required for viral replication. And we identified a number of inhibitors of the nucleoside metabolic pathways. And so we're interested in seeing if these might be repurposed again for use to block SARS-CoV-2 infection. Lastly, I just wanted to highlight EGFR signaling as really important because a number of different EGFR inhibitors came out. So EGFR, as you know, is activated by many different cancers. And so that's why we have so many inhibitors. And so in red here, you can see a large number of inhibitors that we identified as active to block SARS infection. And here are the IC50s as well as qPCR. But see, by employing the second assay, we can see that many of these are not really that potent, but nevertheless, we're interested in identifying the mechanism by which this pathway is impacting infection. And so actually overlaid on this diagram is our RNA-seq data and our proteomics data, which suggests that the infection is really modulating the pathway, altering the phosphorylation status, as well as the levels of many of these proteins, suggesting that the virus is modulating this pathway for its own replication. So again, we're now trying to understand the rest of these hits, and this is a work in progress, but I just want to end by telling you about the lessons we think we've learned and where we're going with this. So as I keep saying, we think there are major differences in cell types. We think the entry pathways are challenging targets, so we can quickly determine which ones are entry and which are not. We're identifying clinically actionable targets, focusing on FDA and those with preclinical activity. We're doing more mechanistic studies, including genomic screening. We're validating everything in the air-liquid interface models and then moving into rodent models. And we're screening additional libraries to really overlap with the data that we currently have. And lastly, we're also doing 2x2 synergy studies to identify drugs that might be synergistic with Remdesivir. So here's an example where we can do 2x2 testing against, for example, the new Merck oral RDRP inhibitor. And we can look to see if we can identify synergistic activities, which really might elevate some of these candidates. So I just want to say that we're really trying to identify antivirals and understand their mechanisms of action and move forward, hopefully, in the future into clinical studies. So with that, I'd just like to thank everyone who's contributed, particularly the High-throughput Screening Core and Dave Schultz, who's the technical director, as well as the other collaborators that I mentioned early on, and my collaborations within Penn. Thank you.
Thank you. Thank you, Sarah. Do we have any questions from the audience members for Sarah? If not, maybe I lead off with a relatively simple question, and that is, are you considering the possibility of testing your positive hits, for example, against other types of coronaviruses in an effort to identify coronapan active reagents?
Yeah, we've screened everything against OC43, and now we're screening against 22.9E and NL63. So yes, we're really interested in seeing if we can identify things that are active more broadly against coronaviruses to aid in potentially the next pandemic, which hopefully won't happen anytime soon. But these viruses seem to jump into human populations with some frequency now. And so we're really interested in seeing if we can be one step ahead.
Steve Goff has a question. Steve, if you can just unmute yourself.
Hey, yes, Sarah, just a quickie. So I was, of course, intrigued by the cyclophilin hit and the apparent action not involving NFAT, maybe. So I wondered, had you tested the old drug that we were interested in long ago, NIM-811, which is another cyclophilin drug?
Yeah, that's active as well.
Yeah, it is. Okay, cool.
Mm-hmm.
And we have a question from Rodney Rothstein. Rodney, you should be able to unmute yourself.
Yes, thanks, Sarah. Great talk. I wanted to know, you chose Remdesivir to look for, quote, double sensitivity, but Remdesivir is such a difficult drug to give to people. Wouldn't it be better to pick a drug that's a little bit easier to give to people for for looking for synergies, et cetera?
So I use Remdesivir as the example because it's now an approved drug, and actually any drug that's now added to Remdesivir has to be tested by the FDA in 2x2 to show that there's no antagonism. But actually we're doing it with a number of candidates that are not yet approved, but are in the pipeline for approvals to do 2x2. We obviously can't do every 2x2 combination because it's exponential, but we are focusing on those that we think will be in the clinic. So that way we can maximize our ability to find something that we think is actionable. But I agree that Remdesivir isn't the only thing.
Yeah. And the other question is, have you started to look at drugs that aren't approved yet, but have targets in the virus? So for example, there's a methyltransferase, and there are some inhibitors against that methyltransferase, et cetera.
Yeah, so we've started to collaborate with some of the groups that have some of the inhibitors, including some new protease inhibitors. Yes, I think direct-acting antivirals, if you can find them, are probably better than host-directed antivirals long-term. But I do think that understanding these sensitivities and pathways that the virus exploits may lead us to new therapeutics as well. But yes, we are starting to work with some other groups that have those drugs.
Thank you.